Hello everyone, welcome to our channel Love of Biology. Today we are going to learn about lac operon model. What is this actually? Gene regulation in the prokaryotes was studied by Jacob and Monod in the E. coli and proposed lac operon hypothesis. And they grew E. coli in the simple medium and provided lactose, a carbohydrate, as a carbon source and observed lactose metabolism in bacteria. So we already learned that transcription and translation as a gene expression. So first let us see this transcriptional unit. Just remember, so the gene which have non-template strand and template strand will undergo transcription. DNA is converting into the RNA with the promoter region where RNA polymerase will bind, and here you can see the structural genes, RNA coding region which contains genes to undergo translation process. After the termination region where the transcription will stop because RNA polymerase will reach the terminator region. So this is the transcriptional unit. So by knowing this, we can learn the lactose. So how lactose is acting on the gene expression, let us see. In presence of the lactose, actually lactose is a inducer in bacteria. So in presence of lactose, different enzymes are synthesized which required for the lactose metabolism. In presence of this, operator will turn on and allow RNA polymerase to undergo the gene expression. So lac operon contains regions. So they are promoter, operator, repressor and structural are the coding regions. Of course, along with this, terminator is present. All these are the regions on the gene. So here you can see I stands for the repressor, P stands for the promoter, O stands for the operator and ZYA are the structural genes. So when lactose is present, the repressor will produce the inhibitor. So this lactose will bind with that repressor and allow the RNA polymerase to reach the structural genes, to move, to slide on the gene and to synthesize the structural regions. It will reach and synthesize the mRNA molecule. Next, mRNA molecule undergo the reaction, will undergo the translation reaction. Yes, and it will synthesize the proteins. Here the enzyme Z gene will synthesize the B galactosidase enzyme and Y gene will synthesize the permease enzyme. A gene will synthesize the transacetylase enzyme. All these genes will help in the lactose metabolism. Beta galactosidase will break down the lactose molecule and permease will permit the lactose to enter. So transacetylase transport lactose into the cell. So in absence of the lactose, operon will turn off and gene does not synthesize these enzymes. So lactose is required for the transcription and translation process. Repressor will produce the inhibitor in absence of the lactose, which will bind at the operator and inhibit the gene expression. So if lactose is absent, gene expression will be inhibited because repressor producing the inhibitor which will bind at the operator and it will stop the RNA polymerase to move. So obviously the gene expression will be inhibited. RNA polymerase can't move, so it will bind. Here you can see at the operator, the inhibitor region, active repressor. So it will inhibit. Thank you for watching. Please subscribe my channel. Thank you.